May peace be on all of you. Myself, Dr. Majid Khan, Associate Professor, Loknethe Dr. J.D. Pawar, College of Pharmacy, Kalwan, Nasik. I welcome all of you to this anti-cancer chapter. In previous chapter, we were cover about the classification of anti-cancer and nitrogen mustard. In that there are alkylating agent, in that there is a nitrogen mustard and alkyl sulfonate. Now in this lecture 2 of anti-cancer agent, here we will cover alkylating agent, in that the remaining subclasses ethylene mines, Nitroso Ureas and Triazine Derivative. Learning outcome: after completion of this topic, student able to understand the activation, mechanism of action and metabolism of ethylene mines alkylating agent, understand the activation, mechanism of action, metabolism of Nitroso Urea alkylating agent, and similarly understand activation, mechanism of action and metabolism of triazine alkylating agent. So now the next class in case of alkylating agent is ethylene mines. The early success of nitrogen mustard led researcher to investigate other compound that contain preformed aziridine ring. Thiotepa incorporate less reactive aziridine ring compared with that formed in maclorethamine. The adjacent thiophosphoryl group is electron withdrawing and therefore reduces the reactivity of the aziridine ring. So during the research of nitrogen mustard, this compound is developed. So here if we see the example is thiotepa, so it is a structure of thiotepa. So here there is a preformed aziridine ring. Previously in case of nitrogen mustard, that is in case of maclormethamine, there is a formation of aziridine ring after activation. But here in case of thiotepa, preformed ring is available. So here already aziridine ring is there in case of thiotepa. Second point here is in case of thiotepa or the thiotepa drug, it is less active aziridine ring. So this aziridine ring which is preformed and it is less active as compared to the maclormethamine aziridine ring. Why? Because it contains thiophosphoryl group. This one is thiophosphoryl group which is electron withdrawing. Therefore it decreases the nucleosity of this nitrogen and thus this preformed aziridine ring is less active as compared to aziridine ring of maclormethamine. Now mechanism action, so it is a reaction of drug with DNA. So here if you see, so it is a drug that is thiotepa which contain aziridine ring. So here the nucleophile which is present on DNA or RNA, it attack to this aziridine ring and there is a formation of complex or called as alkylation. So here directly nucleophile attack to this aziridine ring. So this type of product is formed, that is alkylation. Same reaction is repeated for this ring, then again for this ring. So here alkylation of DNA or RNA occur 3 times. So this straight reaction is related to mechanism of action of thiotepa. Apart from this, there is a side reaction or metabolism. So if we see here reaction, in presence of water hydrolysis is taken place. So here water molecule attack, so there is a breaking of this bond, hydrogen is hydrogen attached to this ring and OH is attached like this. So after hydrolysis, there is a separation of this ring and hydroxyl group attached here. Then this ring after protonation form carbocation, that is nitrogen with positive charge. Again nucleophile from DNA attack to this ring carbon, and this carbon positive charge is neutralized this way. So it is also nothing but alkylation. So after hydrolysis, there is a formation of aziridium ring. Then after protonation, there is a aziridium ring, and then it causes alkylation. Similarly, if below there is an enzyme cytochrome P450, so it causes oxidation. So here there is a sulfur. In case of thiotepa, there is a sulfur, but after oxidation, it's converted into oxygen. So by the enzyme cytochrome P450, this sulfur is converted into oxygen. Again same reaction is taken place as above, that is hydrolysis. Again OH is attached here, ring is separated. Then protonation, it form cation, carbocation. Nucleophile from DNA attached to this ring, and there is a alkylation. So it is the mechanism of action of this thiotepa. So it is a class that is called as ethylene mines. Now next class of alkylating agent is Nitroso Urea. Nitroso Urea were discovered as a result of drug screening by Cancer Chemotherapy National Service Center, which identified N-methyl N-nitroguanidine. N-methyl N-nitroguanidine means this molecule. It is a N-methyl N-nitroguanidine, and this having activity against leukemia means blood cancer. This lead compound was based on the idea that its chemical decomposition was leading to formation of diazomethane, that is CH2N2, diazomethane, and subsequent alkylation of DNA. The activity is further enhanced by attachment of 2-haloethyl substituent on both the nitrogen. So Nitroso Urea compound, and that scientists first discovered that this type of group, that is N-methyl N-nitroguanidine, it is having the anti-cancer effect. It is studied that this compound undergo a reaction and it formed diazomethane CH2N2, which causes alkylation. And further study shows that if we put halogen or halogen substituent on both the nitrogen, so this nitrogen and this nitrogen, if we substitute these two nitrogen with halogen, the activities increases. For example, of Nitroso Urea agent is carmustine and lomustine. So here there is a halogen group on both sides. Similarly, here is a halogen. So insertion of halogen in this type of structure increase the activity. So carmustine and lomustine is an example of Nitroso Urea. Nitroso means NO means nitroso. It is a nitroso. Urea, it is a urea structure, NH2 silver bond O NH2. So here one hydrogen of urea is replaced by this ring, or here is halogen, ethyl halogen. Here hydrogen is replaced by nitroso group, here also nitroso group. So it is an example of Nitroso Urea derivative. Now mechanism of action of this compound Nitroso Urea. So these compounds are reasonably stable at pH 4.5, but undergo both acid and base catalyzed decomposition at lower and higher pH respectively. There are several pathways of decomposition that are possible for these compounds, but one that appear to be most important for alkylation of DNA involve abstraction of NH proton, which is relatively acidic, followed by a rearrangement to give isocyanate and dizohydrazide. The dizohydrazide upon protonation followed by loss of water yield dizo species that decompose to carbocation. So here the reaction is start from deprotonation. So compound, so is a general structure of Nitroso Urea. So here there is a deprotonation. So from this nitrogen, hydrogen is removed like this. So it forms the negative charge on this nitrogen. Then this negative charge converted into double bond. This bond is break, and this bond shifted here. It become N double bond N, then O minus. So there is a formation of two products. The first product is dizohydrazide. So here R, then N double bond CO means it is a isocyanate. So it is a isocyanate, and remaining is that is dizohydrazide. So first step is deprotonation. From this nitrogen, hydrogen is removed. Then this negative charge converted into double bond. This bond shifted here, it become O minus, and then protonation it become OH. So there is a formation of two compound, dizohydrazide and isocyanate. Then this dizohydrazide again after protonation, so here OH converted into OH2. So this OH2, then after removal of water, so how water is removed, this N lone pair converted into bond, so it become triple bond, and removal of water molecule, so it form this diazomethane. So it is a decomposed into diazo species. It is a diazo species after removal of water molecule. Then after removal of N2, it forms the carbocation. So it is a carbocation with plus charge. Then this carbocation is attacked by nucleophile of DNA RNA like this. So there is a attachment of nucleophile of DNA to this carbocation. Second alkylation occur directly, that is nucleophile of DNA attacked to this carbon, removal of Cl group. So there is a dialkylation. So alkylation two times. So once again I repeat the reaction. First is deprotonation. From this nitrogen, hydrogen is removed, it become N minus. Then this minus converted into double bond. This bond shift here, N double bond N, O minus. After that protonation it become OH. So formation of two product, dizohydrazide and isocyanate. Then this dizohydrazide after protonation and after removal of water molecule, it form diazo species. This diazo species after removal of N2, it form carbocation, which causes alkylation of DNA, or there are two types of alkylation. Similarly, this isocyanate also have ability to alkylate the other macromolecule, that is protein. So it causes again alkylation of protein molecule. Isocyanate also causes alkylation of this protein molecule. So it is the mechanism of action of Nitroso Urea derivative. Then there is a inactivation pathway of this drug, that is called as detoxification pathway. Detoxification pathway of Nitroso Urea is also possible, and play a role in resistance to this group of agent. To major route of inactivation first involve dechlorination, which is facilitated by cytochrome P450, and involves cyclization to give 4,5-dihydroxydiazole and isocyanate, which is still capable of carbomoylating protein. The oxadiazole can further degrade by hydrolysis to give several inactive products. So it is a detoxification pathway of Nitroso Urea. So it is a general structure of Nitroso Urea. So here this lone pair of nitrogen converted into double bond N double bond N. Then this bond attack to this carbon Cl minus. So it is a cyclization reaction. Then again here there is a removal of this hydrogen bond shifted to here, and this bond is break. So again there are formation of two products. First product is 4,5-dihydroxydiazole and second one is again isocyanate. Then this oxadiazole further converted into their inactive product, that is several product is formed. So here there is a formation of byproduct is 4,5-dihydroxydiazole and isocyanate. Another route of administration is simple. Here there is a removal of nitroso group. So here simple, there is a removal of nitroso group, so it forms this type of product. So it is a detoxification of Nitroso Urea derivative. Now next class, that is Triazine Derivative. So in that there are two drugs. First one is Procarbazine. Procarbazine is an anti-neoplastic agent that was originally developed as a result of effort to find new inhibitor of monoamine oxidase. Subsequent screening revealed anti-neoplastic activity. It was initially believed that cytotoxicity was related to give hydrogen peroxide H2O2, which in presence of ferric ion produce hydroxide free radical capable of breaking of DNA. Subsequent work shows that although this reaction occur, but sufficient amount of hydrogen peroxide not formed to break the DNA. Metabolism study revealed that oxidation of procarbazine does occur in the liver and is mediated by cytochrome P450 enzyme to give azoprocarbazine. So this procarbazine initially it discovered as a inhibitor of monoamine oxidase, but further study shows that it also has anti-neoplastic activity. And study also shows that the anti-neoplastic activity of this drug procarbazine is due to the formation of hydrogen peroxide. This hydrogen peroxide reacts with ferric ion to produce hydroxide free radical, and which causes alkylation. But hydrogen peroxide so formed is not in sufficient amount to produce this alkylating effect. Then after several years, second study shows that this reaction is responsible for the alkylation. So now see what is this reaction. So here is a drug, that is procarbazine is a main drug. So here by the enzyme cytochrome P450. So here oxidation. So if we see here, there is a NH bond NH. So here oxidation means removal of this hydrogen and formation of pi bond. So here is a pi bond, means N double bond N. Two hydrogen is removed. That is called as azoprocarbazine. So by the enzyme cytochrome P450 or MAO, oxidation taken place. So this two hydrogen from this two nitrogen is removed and formation of pi bond N double bond N. It is called as azoprocarbazine. Then this azoprocarbazine, again the same enzyme cytochrome P450 hydroxylation reaction occur. So here CH2, here hydroxylation occur. one hydrogen is replaced by hydroxyl group like this. So it is a hydroxylation. Then hydroxylation, after hydroxylation there is the formation of two products, aldehyde and methyl diazine. It is a methyl diazine. How it is formed? So this CHOH, so here from OH hydrogen is removed and the bond between OH is transferred here, that is C double bond O, N bond is break, C or N bond is break, so this bond is break. So and this hydrogen which is removed attach here to this nitrogen. So here there is a formation of two product, that is aldehyde and second one is methyl diazine. So it is a methyl diazine. So once again I repeat this reaction. This OH, from this OH hydrogen is removed and the bond between OH is shifted to carbon oxygen. So there is a formation of carbonyl group here like this, and this CN bond is break and this hydrogen shifted to here NH. So like this it forms the two product methyl diazine. So it is the main compound methyl diazine. Either it is converted into free radical, methyl free radical or H free radical, and then this free radical causes DNA alkylation. Secondly, this methyl diazine form the diazomethane. So it is a diazomethane. So here this hydrogen is removed and bond is shifted here, it become triple bond. It become triple bond, that is diazomethane. Then from this diazomethane N2 is removed, only formation of CH3 plus, that is carbocation, and this carbocation causes DNA alkylation. On the other side below, here the first step is same. After oxidation converted into azoprocarbazine, same azoprocarbazine. So again here there is the removal of two hydrogen and formation of pi bond, that is azoprocarbazine. Then this azoprocarbazine after tautomerism. So tautomerism, this CH2 hydrogen from this CH2 shifted to this nitrogen and the double bond shifted here. So it formed this type of product that is called as hydrazone. So here CH2 hydrogen is removed, so it becomes CH, and N double bond and shifted here, it is a double bond. So it is a hydrazone. Now this hydrazone converted into same product, that is methyl hydrazine and this aldehyde, it is same product. So the conversion of hydrazone into this by the mechanism which is as follows. So it is the conversion of hydrazone into methyl hydrazine and aldehyde. So here water molecule attack to this carbon, OH hydroxyl group attack to this carbon, pi bond break, it become N minus, then hydrogen attach here like this. So here is attack of water molecule. Then again this H removed, this bond shifted here, it become double bond, this bond is break and this hydrogen shifted here. So there is a formation of two product methyl hydrazine. So this mechanism is conversion of hydrazone into aldehyde and methyl hydrazine. So it is a formation of two same product. Again methyl hydrazine after oxidation. So methyl hydrazine after oxidation means one hydrogen is removed from this nitrogen, one hydrogen is removed from this nitrogen, and formation of bond, that is pi bond. So it forms the same product methyl diazine. So it is a methyl diazine, same product is formed. Then again this methyl diazine, either it form the free radical or it form the carbocation to causes the DNA alkylation. So here this product is common from this reaction, same product is formed, from this reaction same product is formed. So it is a procarbazine. Then next drug, that is decarbazine. So it is a next drug, decarbazine. Activation of these agents occur through the action of cytochrome P450 to give demethylated product monomethyl triazinoimidazole carboxamide called as MTIC. MTIC full form monomethyl triazinoimidazole carboxamide. Tautomerization allows for decomposition to give amino carboxamide imidazole and diazomethane, which is capable of alkylating agent. So it is the second drug, Procarbazine. So it is a structure of Procarbazine. So here again cytochrome P450 enzyme which causes dealkylation. So here one CH3 is removed like this. So here there is a dealkylation, methyl group is removed. Then in this reaction again by same mechanism that is called as alpha hydroxylation. So here alpha hydroxylation to form this demethylation product and aldehyde. So it is a aldehyde. Then this product, there is a tautomerism. Here also there is a tautomerism. This hydrogen from nitrogen shifted to this nitrogen and this pi bond shifted to here. So like this it forms this product. This product is called as MTIC. MTIC after tautomerization it forms this type of product. And last step, this lone pair of nitrogen converted into bond, that is become triple bond, and this bond is break and hydrogen is attached. So here there is a formation of two product. So here is a formation of two product, that is diazomethane. So it is a diazomethane and this product is called as amino carboxamidoimidazole. Then this diazomethane causes alkylation. So it is a positive charge which causes the DNA alkylation this way. So this reaction is responsible for mechanism of action. Above side, it is a side metabolism. So here first is protonation. So here this nitrogen is protonated like this. So it is a protonation. Then again this lone pair of nitrogen converted into bond, it becomes triple bond and this compound is separated. So here there is a one separated N triple bond N. So it is a N triple bond N. Then after cyclization various product is formed. So after removal of this N2 then it becomes positive charge and after cyclization this product is formed. So it is a metabolism of procarbazine and this one is a responsible for this reaction is responsible for mechanism of decarbazine. So it is a triazine derivative in that procarbazine and tricarbazine. So it is about the alkylating agent. So in alkylating agent there are 5 subclasses. In previous lecture we were cover, that is nitrogen mustard and alkyl sulfonate. But in this lecture or this video we were covered the another 3 classes, that is ethylene in that there is example is thiotepa, then Nitroso Urea derivative in that carmustine, lomustine, and then triazine, that is procarbazine and decarbazine. So these all about the alkylating agent. So in exam the question is asked, solve or short note about the alkylating agent. So it may be for 10 marks or it may be for 5 marks. So if the question is for 10 marks then you have to cover all 5 classes with their reaction, and if the for 5 marks then you shortly write about the alkylating agent of different classes. So in this chapter reaction is important, so you should, all the students should practice this reaction, all the reaction. Alkylating agent in that nitrogen mustard there are various reactions, that is activation of various drug. Here is also there is a various types of reaction, so you practice this reaction. So in this chapter, in case of anti-cancer chapter, all reaction is there. So it is the first class, that is alkylating agent. In next video we will cover the second major class, that is anti-metabolite. So in lecture 3 we will cover anti-metabolite, in that there is a class pyrimidine analog. So the reaction practice is important. So if you like the video then subscribe the channel. Thank you very much.